Thank you. Can you hear me all right back there? Good. My mission is to keep you awake in these few hours we have before lunchtime, delayed lunch. And to start off, I've got a picture of a bumblebee up there, so I don't know if there's any tomato growers in the room. I'm not trying to get you too frustrated, but I just wanted to point out, I can see some good smiling faces. I will be talking about some different animals and different crops potentially that you work with, but what I want to really draw on is the principles that we've uncovered in our study of pollination. So I'm based in New Zealand, but our team, our pollination and apiculture team, works both in New Zealand and Australia. And I think what I really want to, the take-home message I want for you today is just some of the interesting and important facts you can glean about your crops and about how it gets pollinated the more you study it. And so pollination is a fascinating discipline where you have the interaction between plants with their floral biology and their reproductive systems and all the factors that go on in the growing process of the plant, with animals that then have their own behaviors. And not only just a single pollinator species, you might have multiple pollinator species that actually interact with each other. And your pollination is dependent on this interaction, both with a suite of animals and also with the plant. But not only that, both the plants and the pollinators are also responding to the environment in which they live. And it's important to keep these three different aspects in mind when you're considering pollination. And so today I want to give you three stories, and they're just going to be scratching the surface of the field, and there's a lot more data that goes behind these stories. But the first one is some work where we've uncovered unexpected pollinators, animals we didn't think that were playing an important role in pollination. And then I want to talk about how the environmental conditions can actually affect your flowering system, which in turn changes the picture of which pollinators are important. And then lastly, I want to take an example of a pollinator, the bumblebee in New Zealand, and show just a little glimpse of some of the things that we can do once we understand which pollinator is important, things that we can do to develop new techniques for managing that species for pollination. So first of all, unexpected pollinators. Now, I'm sure you're all aware that the honeybee is by far the most important crop pollinator all around the world. And this is because you can move vast numbers of these pollinators en masse into your orchards at will. You get hundreds of thousands of these insects into your fields, and there's no other pollinator around the world that's like that has that flexibility in that number. But on an individual basis, honeybees are not always necessarily the most effective pollinator. Honeybees are a highly evolved social system, and individual bees tend to have a very strict idea of what sort of flowers they want to visit. So if you want hybrid seeds produced from your vegetable crops, or if you have a fruit crop that requires cross-pollination between cultivars, you often find that honeybees are not the most effective at cross-pollinating. And really why they're important is because you can get such vast numbers in there. And an important assumption that we had when we started our work in Australia was that we know that there's managed bees out there, but more importantly, Australia is the last place on Earth that doesn't have the varroa mite. And varroa mite around the world, wherever it arrives, leads to pretty much the loss of feral honeybee colonies. Whereas in Australia, we know that there's a huge population of feral honeybee colonies that live out there in the bush. And it's widely believed that this feral honeybee population provides this free pollination service to Australian crops. And a lot of crop industries will reference this and say, well, this is the pollinators that we rely on, the feral honeybees in their service. But we wanted to look in detail and really understand who was pollinating the crops, both in our fields in Australia and New Zealand. And we want to pick the right pollinators. We want to say, let's get the pollinator that's doing the job, whether that's honeybee or another species, and then we know how to manage it. Because there is a lot of different species that can be visiting the crops. These can be bees, beetles, flies, moths, and there is generally in science accepted that a diversity of pollinators leads to a benefit for pollination. But that doesn't really help a grower. We really need to know who's doing the key role. Once we know which of these species plays a significant role, then we can target measures to manage that particular species, and hopefully that also protects other species too, but we want to target those species that actually do the work. So the standard methods we use when we're evaluating pollinators. First of all, we want to know how efficient they are. For each visit of a pollinator to a flower, how much pollen does it deposit? So we do this process where the top picture there, we bag the flowers before they've opened, and so we have virgin flowers inside that bag. We then take them out and we present those flowers when they're open to a pollinator. So we get this one visit of a pollinator to a flower, and if we do that, build up a big sample, we get an impression for each different species. What's the average amount of pollen they would deposit in a single visit? But that on itself is not enough. We need to know the abundance of that animal in the landscape. If it's a really abundant species and it doesn't deposit much pollen, that may not be too much of a problem because it's just sheer numbers. But other species may not be very common, but each visit they deposit a lot of pollen. So these two things are really important for understanding which are the important pollinators in your crop. But it doesn't stop there. This is an example from hybrid seed production for radish, and I apologize that that photo is the same between the fertile and the sterile line, but just imagine it's a different one. So you'll know if you're involved in hybrid seed production that you have a male fertile line that produces the pollen, that's the donor pollen, and then you have the sterile line, which doesn't produce pollen, and that's the line that's going to produce your hybrid seed. So what you care about is pollination on the male sterile line. You don't care about pollination on the male fertile line. And that's a really key point. And so when we look at some data that we've collected from hybrid radish seed production, on the bottom here, we have 5 different species of pollinators, 2 bees and 3 flies. And then on the y-axis shows the number of pollen grains they deposit per visit. So straight away you can see here that honeybees and bumblebees by far deposit the most pollen every time they visit, and those flies deposit much less pollen. But the important point to note here is this is for the male fertile line, which we don't care about. We care about the male sterile line, which are these white bars. Now, they're depositing a lot less pollen than they deposit on the fertile line, but the trend completely changes. Now that fly species, Aristylus tenax, the drone fly, is depositing nearly twice as much pollen as the bees do. So when you really understand the floral biology and the needs of the system, sometimes your identification of who's the key pollinator can completely switch. And in this case here, even though they're not depositing much pollen, that may be more than sufficient pollen to achieve optimum pollination for your crop. And then when we came to Australia, we were looking at avocado pollination in the tri-state area around Mildura. I just want you to ignore the graph for a second. Again, our assumption here was that there's this feral honeybee population that's pollinating the crop in Australia. And now this graph here shows at the 19 different orchards we were looking at within this region, within this tri-state area. This is a standardized index of abundance of these 3 different groups of pollinators that we found in the orchards visiting the flowers. Honeybees, flies, and beetles. Honeybees are the blue bars, which you can hardly see. There were some orchards where we had no honeybees visiting the flowers at all. And the orchards where we were having honeybees visiting the flowers, they were visiting at much lower frequency than the flies. Now, this is important because these growers in this area assumed their crop was honeybee pollinated. Most of them didn't bring honeybees into their crop, but they assumed there was honeybee pollination from the feral honeybees that were around. There was one grower who brought 2 hives into his orchard, but not much. But actually what it turned out, this is just the abundances, but we did the study to demonstrate that in actual fact it was these flies that were pollinating their crops. And so although there was an assumption out there that the crop was pollinated by the feral honeybees, it turned out in actual fact that in this region, the avocados are pollinated largely by flies, and those flies are doing a great job. The important point to note is there is bees in this environment, but these orchards are surrounded by citrus orchards. And the bees are all over the citrus flowers, and they simply weren't coming into the avocado flowers. And yet those flies were sufficient to do the job. So that just gives you a sort of 2 examples there of how it's important to understand who is actually visiting your flowers and what they're doing. But then when we bring the flowers into it, the situation gets even more interesting. And I'm going to give the example here from avocados again. Is that going to work? Okay. So avocados have a really interesting floral system. Each avocado flower opens first in a female phase. And this is a female phase flower there with a stigma sticking up. That female avocado flower is only open for about 2 to 3 hours in the morning on the first day. That flower then around midday starts to close again, and you'll see the flower will start to close, the anthers will curl up. That flower closes and opens on the afternoon of the next day as a male. So across a whole tree, you have a sequence of female flowers in the morning, closing at midday, and then these male flowers that start to open up in the afternoon. And what's more, if you watch and you wait as this female flower is closing, this male isn't producing any pollen yet. You have to wait until these anthers open up, and in a second you'll see these flaps starting to open. When those flaps open, that's when pollen's being released. And by that point, the female flower on that same little branch there, you can see the flap has just popped up. That female flower is now completely closed. And this is the Hass avocado. Different cultivars have different patterns, but generally it's well known around the world that Hass female flowers open in the morning, close, and are male in the afternoon. So every avocado that you eat, that avocado came from a pollination event from a flower that's only open for a 2 to 3 hour window. It's a very tight period of time for pollination to occur. What we know from the literature is that this standard pattern sometimes changes, and what's generally sort of reported is that at colder conditions, the pattern switches, and the female flowers opening in the afternoon. And we wanted to know more about this. So for 3 years now, 3 years, we've collected data, detailed data on this flowering cycle as it relates to temperature. So we had these cameras taking time-lapse photos every 5 minutes associated with weather stations so that we could really understand in detail how temperature affected the flowering. And this is some of our data. Now, on the bottom axis there is the overnight minimum temperature. There's lots of other factors we could look at, but if we just look at the minimum temperature it got to the night before flowering, as the temperature drops, the time at which the female flower first opens is shifted later and later in the day. So essentially, we break it down into these 3 sections. In the red section, these flowers are performing as normal Hass flowers are said to perform. They open around 10 a.m. in the morning, and they close around midday. But when you get into the green section, between 9 degrees and about 10 degrees Celsius overnight minimum temperature, the flowers actually open at midday and closing in the early to mid-afternoon. When it gets colder again, from about 4 degrees Celsius up to 7 degrees Celsius, those flowers are now opening in the late afternoon. And the interesting thing about this graph just shows opening time. This group of flowers, those female flowers, stay open into the dusk all through the night and don't close until about 9 or 10 the next morning. Now this is a crop, I don't want to get too much into the details about avocado. The important point is this is a crop where we all knew that the flowers closed at nighttime. The flowers were only receptive in the morning. And here we found that in actual fact, in a significant proportion of the nights in our season, the flowers were open all night long. And what's more, there was a lot of things visiting. So here's a picture of a noctuid moth that's going carefully from flower to flower on these female flowers at night. So not only were these flowers open at night, suddenly there was a whole new suite of pollinators we'd never previously considered. And when you speed up the footage, it reveals even more. There's a whole lot of mosquitoes, fungus gnats, crane flies, moths, and other bugs that are visiting these flowers at this nighttime. And there's a whole lot of work to try and assess their role as pollinators, but this just shows the importance of understanding your flower, understanding how it responds to environmental conditions, and what that means for the important pollinators. We've carried this work on with other crops that don't have a visible closing at dusk, and we've found cryptic patterns of receptivity, which mean that sometimes the crops are actually more receptive in the evening than they are during the middle of the day. And it's important to understand this for your crops so that you know which are the pollinators that are visiting the crops that are important. Once you know your pollinators, once you know who's important, then you can really look and say, can you develop targeted methods to benefit these pollinators? I was going to talk a little bit about the bumblebee, which is a very effective pollinator. We'll be making a contribution to pollination in Tasmania, but don't tell anybody. What we are trying to do is develop these bumblebee bunkers, which are artificial nest sites, to try and have a really practical means for growers to manage these colonies on their orchards. So bumblebees have a very different cycle to honeybees. Honeybees last from year to year. Bumblebees, in summer, you have a big colony that's developing. At late summer, that colony starts to produce queens and males. Those queens and males mate in autumn, and then the whole colony dies off, apart from the young queens who have just mated. Those young queens overwinter in holes in the ground, and then they emerge in spring. They start foraging on nectar and pollen and build a nest from scratch. And so we really wanted to target that nest initiation phase. We wanted to build an artificial nest site that those young queens emerging in spring would prefer over a natural nest site so we could get them into our bunkers. So we've had a program now for 4 years doing this artificial nest site. It's turned into a big program. We've got over 1,000 of these bunkers in 5 regions in New Zealand, and from avocado orchards to red clover fields and plum orchards. And I'm not going to get into the details of what we did in this program. But there was one really important result that came through from the very beginning of our work. There's 2 main species of bumblebees that are common throughout New Zealand. The short-tongued bumblebee is by far the most common species. It's more than 20 times common. In some of our surveys, we found more than 40 times common than the next species. And yet, this most common species only turned up in 3% of our bunkers. In contrast, long-tongued bumblebee species, and there's 2 species here, they are by far much rarer than the short-tongued bumblebee, and we had 12% of our bunkers had colonies of this species. And this pattern has been repeated in all the 4 years of our study. So what this suggests to me is that we have a bunker that works well for the long-tongued bumblebees, but we don't have one for the short-tongued Bombus terrestris. So what is it that these Bombus terrestris are looking for in a nest site? If we can figure that out, we could change pollination in New Zealand. If we had the same success rate with these common species as we do with the rare species, we could really recruit these as pollinators in our orchards. So now we've had quite a lot of work going into understanding the behavior of these bees in detail. And to do that, we've had to go to some pretty extreme means. So we've been using little active radio transmitters that we glue to the back of these queens at that stage where they're initially establishing a nest site. And what we do is we catch the bees when they're collecting pollen off a flower, we put the transmitter on, and then we can track that bee back to their nest site. Once we track them back to the nest site, we can understand the characteristics of the nest site at that very early stage. So we can say, what kind of soil is it in? What is the tunnel dimensions? What's the cavity look like that they're nesting in? These tags are 200 milligrams in weight. These bees are between 600 milligrams to a gram in weight. So this is a pretty big package for the bee to be carrying. So we've also been doing quite a little bit of work to understand what are the effects of putting a transmitter on a flying insect like this. So we've built this flight tunnel where the bee thinks it's flying forward so we can get good, consistent flying motion. And we use a high-speed camera then to look and understand how the addition of a transmitter changes the frequency at which it beats its wings, the extent of each wing beat, the angle at which it holds its body. And we're able now to really understand in detail the effects of putting the transmitter on. And I think this is important because we're paving the way here for the use of these sort of transmitters in a much wider range of animals. We've got a project that we hope to get started this year where we're actually planning to reduce the size of this radio transmitter from 200 milligrams. At least our aim is to get by an order of magnitude down to 20 milligrams. And the important thing about these radio transmitters is they release a radio pulse. We can detect this pulse up to 1 kilometer away. This is not a short-range transmitter, so it provides you ecological scale data on the movements of these animals. So we've been using this to understand the selection of nest sites by bees in the wild, using manual tracking methods where we have the transmitter and we track it down. And this is from our field site in the Netherlands with my collaborator from the University of Amsterdam and my master's student there. But the problem with manual telemetry is once a bee flies out of your study site, you've lost it. You don't know when it's going to come back. It's a huge amount of work to track these bees. So we've also developed an automated telemetry system where we can track over an area of about 90 hectares. We can track the movement of up to 200 individuals simultaneously, 24/7. We can see all their movements within the study site. And this gives us unprecedented ability to understand the movement of these species through an environment. And so you can imagine, if we can do this for queen bumblebees now, if we're able to reduce that transmitter even further, to look at honeybees, or here in Australia, there's many native bee species that could potentially be used as pollinators. If we can use a system like this, we can understand what the nesting requirements of native Australian bee species are and develop the ideal nesting habitat or the ideal nesting boxes to utilize these species for pollination. So I want to leave you with a few questions to consider about your crop. You may not grow avocados or some of the other crops I've talked about, but do you really know who's visiting your flowers? And are they moving the pollen in the right way? Are they doing the cross-pollination that's required to get the high-level optimum seed set? And do we understand enough about the flower and when it's receptive, those cryptic patterns of receptivity, to know that the pollinator that you're looking at is visiting at the right time for the flower. And once we've identified those key pollinators, what new tools can we use to understand their behavior and to develop techniques to manage those pollinators to improve pollination? So thank you.